Hey kids, it's week 3, lecture 1 on bioinorganic chemistry. So today's lecture is going to be relatively short. I'm going to talk a little bit about the history of the field, discuss briefly some biological ligands and why they're important, what I think you should take away from that. And then we'll look at metals in biology and see how they're used in general features. We'll then dive more deeply in future lectures during the week. Okay, let's think about the history of the field. An enzyme was defined as a biological catalyst in 1833, at least that's when they were discovered. And we didn't know what enzymes were for a long time. Jack bean urease was discovered and isolated and studied by James Sumner in the 1920s. And Sumner actually proposed and hypothesized that the protein urease was the catalyst. It actually took quite a bit of time for that to be accepted by the scientific community, but it turns out that enzymes are proteins. This leads to the quote at the beginning of the chapter, which I'll read. In 1926, Sumner isolated from jack beans the first crystalline enzyme, urease, and defined the proposition that enzymes could be proteins devoid of organic coenzymes and metal ions. It is therefore with some sadness that in this communication we adduce evidence which strongly indicates that urease is a nickel metalloenzyme. Not only is it a metalloenzyme, but that nickel is required for the protein function. So just like in the introductory PowerPoint where I showed the carbonic anhydrase, which used the zinc, the urease enzyme includes nickel, which activates a water molecule for chemical reactivity. So here is a cartoon of the active site of urease, and what we have here is nickel 1, which holds onto the substrate, which is urea, and the urease enzyme breaks down urea by having a hydroxide attack the urea. Hydroxide is not strong enough to do this reaction, and so a base that's present in the active site deprotonates this water molecule, which has already been activated by nickel 2. We form this tetrahedral intermediate, which is stabilized by the protein environment. A donor from a sulfur protonates the ammonia. This carboxylate is there to help stabilize the positive charge and sort of hold the urea in place. And so then we end up forming this product, which has lost an ammonia, and the chemical reaction has taken place. So the enzyme requires both nickels working in a concerted fashion to cleave the bond in urea. Let's talk briefly about the unique fitness of transition metals for bioinorganic chemistry. The first is that transition elements, unlike the p-block elements, can exist in multiple oxidation states. This allows them to be redox active and allows for much of the redox chemistry that we can see in biological systems. That's not to say that you can't have any redox without a transition metal. So for example, tyrosine can be oxidized to the tyrosyl radical. But in general, it's the oxidation states of especially iron and copper, but other elements too, that allow you to have redox chemistry. In addition, so transition metals have d orbitals and some d electrons, and that allows, what's going on here? Well yeah, if you're studying Gen Chem, you have one way of viewing this, but inorganic chemists view electrons and orbitals slightly differently. Oh, it's going away. I guess that's the end of that. So in addition to oxidation states, metals can have multiple coordination environments. And this is best seen in copper. Copper 1 has one geometry. Copper 2 has a different geometry. And the homework this week, one of the homeworks, is having you look at prove this claim that copper 1 should be tetrahedral and copper 2 should be square planar. Enzymes can use this behavior to actually stabilize or promote the coordination environment of this copper in this example, which has neither a tetrahedral or a square planar environment, but a geometry that's very close to a trigonal pyramidal geometry. So here's the copper in the center. It's bound by 2 nitrogen histidine ligands and 2 other groups that I'm totally blanking on right now. I think there's sulfurs and methionines. And this copper is held in a position that it doesn't really want to be in, and that is causing this copper to be a redox center that has a lower activation barrier. And this is the entactic state principle, which is it's described in detail in the textbook, but what I want to just emphasize here is if this is the reaction coordinate for the redox chemistry of copper 1 and copper 2 in just aqueous media, the copper has been destabilized at the starting and at the ending point. I realize I'm covering up the word there. So it's less stable as a starting material and as a product, and so the delta G of activation for this process is lower, meaning the reaction is faster. And so the tuning of the sterics by the protein environment, the specific geometry of this copper ion, is tuned by the protein environment. So this copper is neither tetrahedral or square planar, but it's in an intermediate geometry, and the protein environment holds it in that orientation. So protein environment is going to be very important. The protein structure is very important. For a biochemist who may be very interested in studying proteins without metals, then the protein structure is its own thing. But for the purposes of bioinorganic chemistry, and this is a gross simplification, the purpose of the protein structure is to provide a coordination environment for a metal ion. Certainly there's more to it than that. When we get to our case study in the final week of the semester, when we look at nitrogenase, we'll see that the protein environment provides pathways and channels and protons and electrons and all these other things, but the main thing that it's going to do for the purposes of this module is to provide an environment for metal binding. So we need to have a rigid structure and a very precisely ordered structure. And so this first type of protein structure is known as the alpha helix. The alpha helix is a regularly repeating arrangement of amino acids that spirals as it moves up. And it's usually drawn as a ribbon diagram to emphasize sort of the backbone without the amino acid side chains. So you can see that it forms a nice circular arrangement. The repeat is approximately 3.2 residues, so there's not exact overlap as you move up. It's about 3.2 turns per 3.2 amino acids per turn. So, but the alpha helix allows these amino acids to be pointed in various directions around the chain, and it's a fairly rigid structure. There's a lot of hydrogen bonding within this helical coil which holds it in place. So the alpha helix is used to provide structure and position amino acids in the protein. The second major structure, and the only other one that we'll discuss, is the beta sheet. And the beta sheet is a long chain of amino acids that goes one way, and then there's something else up here, and then it comes back this way, and then that stuff down here, and then it comes back this way. And so you can have beta sheets that alternate, so up and then down and then up. You can have them where they're parallel, but the way they're drawn in the protein is like this. And this is, again, another type of a structure. It's held in place, again, with hydrogen bonds between the strands like this that holds it in place in a fairly rigid way to orient amino acid residues in a preferred orientation to do whatever it's going to do. But for this class, it's really most important for positioning amino acids to bind to the transition metals. The last type of protein structure is random coil. We're not going to talk really a lot about that, but anything that isn't a structure, so what's out here, it might be some random coil that it doesn't have a defined structure but is present. The main reason why we use these ribbon diagrams is because this is much easier to visualize than this. This, atomic resolution of the protein environment is how we typically think of chemistry, atoms as being little, spheres of electron density. And the problem is it's too much. The biological proteins, the metalloenzymes are just too big to really visualize. And the main thing that we care about is this heme group. So a heme group is an iron contained within a tetranitrogen planar ligand, a porphyrin. And so this is either cytochrome C or hemoglobin. I don't actually remember which picture this is, but this heme group and the iron here is held in a very specific orientation by the amino acid residues that are pointing in from these alpha helices. So the alpha helices provide this structure, this rigid structure that then allows the heme to be in a very specific orientation inside the protein. So I'm going to ask you to think about this as you're looking at the slide, but let me introduce it first. These are the 20 naturally occurring protein side chains found in biology. There are several additional ones found in organisms that are not coded by genetics, but are added in a post-translational modification to the backbone. But these 20 are the ones that we normally think of as being the amino acids in life. So take a look at these side chains and think to yourself for a moment, which ones of these might bind to a transition metal? We've talked a lot about ligands this year. We certainly haven't talked about every possible ligand, but if you think about what a ligand might be, look at these side chains and try to come up with some general trends for which ones would be ligands for a metal. I will pause for a few seconds and then come back. So your first thought might have been the electrically charged groups would be good ligands, and that's not a bad place to start, but it's not the whole picture. So if we think about what the best ligands are for transition metals, by far and away the two most important ones are cysteine and histidine. So cysteine is here, and histidine, I just had it and it's gone. Here it is, histidine. So these two provide sulfur donor atoms and nitrogen donor atoms to a metal. And they're very, very common ligands for transition metals. But other ones are aspartate and glutamate, methionine, and tyrosine. So we can see that they span nonpolar, polar, and electrically charged groups. So depending on what element you are, you might bind to different amino acids in different ways. You could also certainly imagine binding to threonine, which has an oxygen, serine, which has an oxygen, asparagine and glutamine, which have heteroatoms that could bind to a metal, or even arginine and lysine, which have heteroatoms. But in general, cysteine and methionine are definitely the most, histidine. Cysteine and histidine are the most common ligands for transition elements in a biological system. The other side chains that are charged can provide protons or bases. So these can be acidic because they can donate a proton. These are basic; they can accept a proton. And a lot of the nonpolar ones tend to be structural only. So these hydrocarbon groups can provide a steric environment for the protein, allowing it to have a very precise, exact shape that then allows the protein to fold in a certain way, again, pointing these ligand side chains towards a transition metal so it can bind to that metal. I'm not going to claim that that's the only role for amino acids in biochemistry. There's certainly a lot more, but for the purposes of what we're thinking about, getting the ligands pointed into the correct place so that they can have an exact coordination distance and coordination geometry around the metal is going to provide the information that that metal needs to have its specific redox potential, geometry, and chemical reactivity. Now let's think a little bit about DNA. DNA is the encoder for making proteins in biology, but it also, can have some roles for binding metals, which we'll see in just a second. So the 4 DNA bases are adenine and thymine, which form a DNA base pair due to hydrogen bonding that looks like this, and then guanine and cytosine, which form a hydrogen bonding network like this. These hydrogen bonds hold the DNA bases together in the center of the DNA molecule, and then there's the phosphate diester backbone that winds around it. You've all seen pictures of what DNA looks like. It's a nice winding strand. There's pictures in the textbook chapter. There's a major groove and a minor groove. And it turns out if you can disrupt DNA, then you can disrupt protein encoding and cause cell death. And this is the basis for cisplatin, which is a extremely powerful anti-cancer drug. Most anti-cancer treatments use a platinum-based drug, at least at some point during the treatment. And it's because the platinum, when it binds to the DNA, it does so and causes a significant, severe kink in the DNA. So instead of it being straight, the DNA curves pretty dramatically around the platinum. Platinum binds to two of the G bases and puts a significant strain on the DNA. It actually disrupts the ability for that DNA to be translated and turned into proteins. So this can cause death of the cell, and for rapidly dividing cells seen in cancer cells, that's how the treatment works. So part of your homework, one for this week, is to propose a synthesis of cisplatin and transplatin using the trans effect types of arguments that we talked about earlier in the class. The last topic for today is just to think about metals in biology. Metals have a lot of different roles. There's the bulk metals, which are the ones like sodium, potassium, phosphorus, sulfur, halides. These are found in bones and your phosphate diester DNA backbone, also for charge balance, charge carriers in the cytoplasm. So these are present in large quantities in your body. The essential elements are most of the first row transition elements, not all, but most of them. They have essential roles for either structure, redox, or reactivity, and they're important roles known for most of the first row transition elements. And finally, there are those that are toxic, medical, or used as diagnostics. So for example, gadolinium is used as an MRI contrast agent. We saw on the previous slide cisplatin being used as an anti-cancer agent. And the difference between something being toxic and something being medicinal depends on the dose. There's a figure in the textbook chapter, but also on Sakai, which talks about how doses of the metals have a different response depending on whether the metal is essential or non-essential. So looking at the curve for an essential trace element, if there's a deficiency, you can have a severe or a mild response. This graph is specifically looking at plant yield. So if you don't have enough of your element, then you won't have as good of a yield as you'd like. There's an optimal range of concentration that is sufficient. And if you think back to the introductory video that I posted before this one, the amount of concentration that we have to do relative to what's found in the natural environment is pretty severe, from maybe 20 to 20,000 times concentration. So you want to have the very precise range for each specific element. But then if you get too much, if you have above the critical concentration, you can then begin having toxic effects that can be mild or severe and can cause the death of the organism. For a non-essential trace element, if there's a low concentration of the element, there's no real effect, but as you get above that critical concentration, then you can have mild to severe effects. And again, this graph is showing plant yield, so plant death as you increase the amount of toxic metal ion in the environment. Okay, so that's it for this lecture. The next lecture will cover various cofactors, hemes, iron-sulfur clusters, and zinc fingers. We'll talk a little bit about these interesting structures and what they do.